In general, if we talk about the field of tissue engineering, it encompasses many, many different aspects of science, engineering, technology in general. So it's extremely difficult to attempt to capture this in 4 short 10 to 15-minute talks tonight, but we'll do the best we can to give you some of the essence of it. And that's really how we've organized the four venues. One will be David Scadden, who's at Harvard University. David will tell you something about stem cells and basic stem cell biology and the role of stem cells in regenerative medicine in general. I will then give you a view into one area of biomaterials and the role these kinds of materials play in the process of engineering new tissues in the laboratory. Then Gordana Vunjak, who's at MIT, will give you a talk about one particular type of tissue engineering product, if you will, one of the organs or tissues she's growing. But I think she'll also introduce you to some of the other tissue types at the same time, but she'll focus on her heart tissue engineering. And then finally, Mehmet Toner, who's at Harvard, will give you a perspective of where biology and technology need to come together if we're going to make some inroads, not just in tissue engineering, but in the broader field of medicine as well. So the talks are purposely a bit broad to give you that perspective, and we hope you'll get engaged in questions, discussions, both with ourselves and with our postdocs and students who are here and eager to talk to you. Let me also acknowledge, first of all, the National Institutes of Health. Not only are they instrumental in keeping all of our labs and students funded and eating at night, but also for hosting and for providing the support for things like the conference going on in Boston that I'll allude to in a second, and also very important to Gordana and myself in running what's at the bottom there, our Tissue Engineering Resource Center. So these are important venues where your tax dollars directly then support the kinds of research you'll hear about today, and hopefully will have a major impact over the coming years in everyone's health and well-being. The reason we chose this particular date is simply because over the course of these 3 days, 2nd, 3rd, and 4th, over at the Cambridge Marriott, we've organized a 3-day workshop where we're looking at this whole field of tissue engineering and asking questions about where the field needs to go in the next 10 to 20 years. And so out of that process that's going on down the river, we'll hopefully come up with a set of guidelines that will be very important for all of our research activities, both in terms of fundamental and applied issues.
A little bit of background about me. I'm a physician. I'm a hematologist oncologist, and so I actually work with stem cells that we use in clinical practice on a regular basis. And I'd like to give you a sense of really the basic aspects of stem cell biology that make these such a very powerful tool that we think will really transform medicine in the future. Now, stem cells mean a lot of different things to a lot of different people. I particularly like lower right, but I think probably not achievable in our lifetimes. But one of the things that is critical that we all have the same language, and I think as Daniel Patrick Moynihan once said, we're all entitled to our own opinions, but we're not entitled to our own facts. So I thought I'd give you a little bit about some of the issues of what are the facts of stem cell biology. And one of the things I want to go through with you is the fact that we do currently use stem cells. Again, this is not something that is just a futuristic therapy, but is something that is a very important part of current medical practice. It's used primarily in the treatment of people with blood diseases and cancer, but it is now extending out and involved in the treatment of heart disease, and also it's critical for some of the areas of tissue transplant, in particular skin grafts and corneal transplants. Now, it's important to recognize that while this is a common practice now, there is a history, a legacy of controversy that even attended this area of adult stem cell research that we now think of as being accepted and unproblematic. And that is adult stem cells, when first used, were thought to be something that were somewhat scientifically suspect and were ethically of great concern. The idea that you could create people who would to some extent have the parts of other people within them was very troubling. The idea that you might be giving cells to a person that were derived from someone else and could potentially, it was thought, harm that person, harm the donor, was also something that was very controversial. So I recently had a wonderful experience in that The person who did the first solid organ transplant 50 years ago was in the audience at one of the stem cell talks and came up to me afterward and said that, when he had first done that kidney transplant, it was done at the Brigham here in Boston. And when he was doing that, he said that it was just there was just a maelstrom of criticism and concern. And it said it's very similar to the current stem cell debate, that it's an area where when you're exploring something new, nobody really knows what's going to happen. And at some point, you just have to do it. You have to try it, and you have to be courageous and put up with and convince people that it's worth taking the risk. And from those early experiments, he went on to both win the acknowledgement of the scientific community through a Nobel Prize, but he's clearly changed human health. I mean, we now have the ability to transplant organs, and that's just made a huge difference and saved tens of thousands of lives, hundreds of thousands by now. And it also actually spawned a whole area of science, and that is modern immunology, which has had a huge impact in many different areas, and something that is a palpable part of clinical medicine on a regular basis. So just keeping that in mind, I want to go through with you some of the issues of stem cells, both as a general topic, the issues that differentiate the kinds of stem cells that are raising a great deal of ethical concern and political debate, the embryonic stem cells, and then talk about the adult stem cell, and talk a little bit about why we need each of these. And then I'll end it with an issue related to one of the most controversial areas, and that is cloning. Now, all of this, I haven't mentioned the word yet, tissue engineering. And I have to tell you that the reason that I think I've been asked to speak this evening is because as we think about ways in which we can make the constructs that we envision tissue engineering to be very powerful in the future, that they will need to be populated, that these bioengineered devices will need cells that populate them. And those cell types, if we just use adult cells that are more mature cells, they will have a short lifetime. They exhaust themselves. It's the stem cell that provides the resource that is self-replenishing and will allow for the sustained production that will maintain these tissues long term. So this is an indirect way of leading into the topic of tissue engineering, but a critical component if we think about the future success of tissue engineering in clinical medicine. Okay, so what is a stem cell? A stem cell has two very characteristic properties, one of which is that it by itself doesn't do a whole lot. And I'm having a hard time seeing without my glasses. It's that one. Thanks. And basically, it by itself is a fairly nondescript population of cells that's generally relatively rare in an adult. It's a population, though, that gives yield to all of the different mature cells that make up our body. So these are the real business cells of conducting the functions of different organ types, but they have a relatively short lifespan. So, for example, certain blood cells only last 7 hours. If we had to, so we therefore require a whole population of cell types that's capable of generating massive numbers of these. The number of blood cells that you need to be produced every day just to maintain normal blood cell numbers is 10 billion. So the production of cells is enormous. And it occurs mostly at these progenitor populations, which have the capacity to expand and proliferate gangbusters, whereas up at the top of the heap is the stem cell that really does very little other than occasionally turning over and making a new one of these. If it was a one-to-one relationship, we would obviously exhaust these cells very quickly, and so there actually are relatively small numbers of these. These grow very, very rapidly and make, again, all of those mature cells. But even so, their slow division would result in their exhaustion unless they're capable of making more of themselves. And the beauty of a stem cell is that it's capable of doing both, that it has the ability to produce all of the offspring that are the mature cell types, as well as replenishing itself. And so it is this balance that allows us to be able to continue to produce and renew the new surface of the skin or the lining of the mouth or the blood, the bone marrow. And all of the different tissue types seem to now have been identified as having primitive populations of cells that are capable of making more of the mature offspring. Now, the beauty of these cells is that they have then the capacity to possibly make all different kinds of tissue types and are therefore thought to be a very strong resource for creating new types of cell-based therapies, and I include within this bioengineered devices that may contain many different cells in particular ways of organization. But I think it would be wrong to view stem cell biology as simply the production of replacement parts, because there are ways in which we will be able to use this new information to understand diseases that we don't currently have any good models for, and to be able to develop new drugs for them. In addition, as we learn more about the stem cells that exist in most of the adult tissues, we hope that this will lead to the development of new drugs for tissue regeneration. And then finally, I think the area of stem cell biology will be very important in providing new insight into the mechanisms that cause cancer, and we hope will change the way we do cancer drug development. So while this is perhaps the most easily understood way in which using cells can provide therapies, that is, that we can basically replace damaged tissue, there are a whole range of other applications that I think will change the face of medicine over the course of the next 10 years. Now, just to go through what different stem cells are, the cells arrive at different times in development that have very different properties. And these cells have the capacity of producing new cell types based on where they were derived in the course of development. So stem cells that exist early on, immediately after fertilization, are those cells that, after the first cell divisions of the fertilized egg, if they were to split apart, could form an entire new individual. They can form both the embryo itself and all of the surrounding tissue, and so those are called totally potent cells. Now, during the first 5 to 7 days of life, the cells are continuing to divide, and they become a structure that's a ball. It has about 200 cells in it, and it starts to have this structure to it. So it has a little cystic area, and then a collection of cells at the top that's called the inner cell mass. Now, this is a collection of cells that's about the size of the sharp end of a pin, and has not yet gone from being a fertilized egg to a structure that's embedded into the womb of the mother. It doesn't have any viability unto itself unless it does embed in the uterus. If we take those cells, and if we take that structure and isolate the cells that make up this inner cell mass, these cells, if you grow them on a plate, can potentially be grown in a way that allows one or two of them to start to expand. That population of cells is called an embryonic stem cell line. Now, the way these lines are usually derived is that as if a couple goes to a fertility clinic and they go through the process of fertilization outside the body, the cells begin this division process, and they get to this point, and this embryo could then be implanted in the womb of the mother and result in the generation of a child. However, there are often excess embryos that are created. Those embryos can then be maintained for a period of time in the freezer. However, if a family decides that they're no longer going to continue to have children, this then becomes a dilemma as to whether or not these should be discarded, maintained, adopted, or donated to research. With the recent early passage of legislation on Beacon Hill, it may be possible now for us to be able to use these in research in a way that would allow us to create these embryonic stem cell lines. These lines have abilities to become all different tissue types. They are very different than the cells that are derived if you continue to allow this to differentiate, and this would, of course, now only occur in the setting of the uterus. It could go on to then form a fully formed individual. Cells derived from either the fully formed individual or from the umbilical cord blood that is present at birth are really multipotent cells. And they are loosely called adult stem cells or somatic stem cells and have very different properties than the embryonic stem cell. And I'll tell you a little bit about how those two are distinguished. So the embryonic stem cell has very unique characteristics in that it is capable of self-renewing on a very regular basis. So these cells turn over and make new cells about every day and a half. In addition, they have the capacity to become specialized cell types that are of many different types. They can become things like blood, they can become things like the nervous system, or the pancreatic islet cell that's important in insulin production. That's critically important to prevent diabetes and treat diabetes. So the idea that this cell then is something that is capable of turning over very rapidly so we can make lots of it and has the capacity to become all these different tissue types is something that has made it a very powerful, compelling tool for research and we hope ultimately for therapies. Now, the complicated aspect of these is not just their ethical dimension, but also the fact that when we grow them, we know we can maintain them by putting them in the context of a particular type of medium. When that's withdrawn, what happens is the cells spontaneously become a range of different specialized cells. And some of those specialized cells are very interesting. They become things like a heart cell. And unfortunately, in transferring this over, I couldn't show you the fact that a group of the cells over in this corner will actually start to beat, which is something that late at night looking under a microscope can really get your attention. And they have the ability then to really to become many different tissue types, some of which actually resemble something like a mature heart cell. But unfortunately, we don't have the capacity at this point to be able to drive this in a directed way so that if we want heart cells, we get just heart cells. Or if we want just those insulin-producing cells, we get just those cells. Unfortunately, in this collection, we can often find cells that are not just heart cells, but maybe some blood cells, maybe some cells that are going to become skin over here, maybe some nervous system cells over there. So it becomes it is still a very complicated process for us to be able to sort out what are the cues that drive the growth of these cells or their specialization in ways that we could ultimately we can ultimately direct it. So we still need some very basic tools to be able to use these in an effective way in therapies. But the advantages are that they can become all of these different specialized cell types, that we can grow them, and we can grow them basically indefinitely. And because we can grow them, they are actually cells that we could potentially subject to a gene correction. So you might imagine then, in the setting of someone with a genetic disorder, that it might be possible to alleviate that disorder by providing a new gene, and in that way provide them tissues that may not be damaged by a genetic abnormality. There are lots of good things about these cells, but clearly there are also lots of cons, including the ongoing ethical and political debates. The fact that we really don't understand how to direct them in a very organized way. The fact that they can become all of these different tissue types is actually one of their weaknesses as well, in that if you put these cells into an animal, they actually will form a tumor. They'll form a tumor that's a disorganized group of cell types. It's called a teratoma. And because these necessarily come from an individual embryo that will not mature, they are necessarily different than the possible recipient of these cells. And so the immune system would see them as foreign. I think I'm going on way too long here, but let me just tell you briefly about adult stem cells. These are cell types that we have recognized as being present in the bone marrow and capable of being able to basically transfer the capacity to form all of the blood elements and immune cell elements with a simple transplantation. And I say simple, it's a very complicated process, but it's one that can be done in a laboratory setting with just a single cell. Now the problem is these cells tend to turn over very infrequently. So unlike the embryonic stem cell that turns over about every day and a half, the adult stem cell, particularly the blood stem cell, has been characterized as turning over on about an every 6 to 12 month basis. So they don't turn over very frequently. They're very rare, very hard to isolate, and very difficult to work with in a way that can allow us to manipulate them very readily. In addition, what they can become is very narrow relative to what the embryonic stem cell can become. So if they are a blood stem cell, they can make all the different cell types of the blood, but they really can't go on to form brain or the islet cell of the pancreas that produces insulin. So it has been appreciated that some of these, with very rare frequency, can possibly become other tissue types. But for the most part, those events are too infrequent to be useful clinically. So these are generally regarded as just multipotent, as having a very narrow band of being able to produce a particular cell type. So if you want to get a cell type that's going to form a new brain cell, you need to get it from the brain and be a neural stem cell. If you want to make blood, you get it from the bone marrow and have it be blood cell, stem cell. So markedly different than the embryonic stem cell type. Okay, now these are terrific because we already use them clinically. We know that you can get them from the individual, so you won't have this problem of immune rejection. They do have this ability in the clinic that has been demonstrated in a number of different settings, but they're rare cells, they're hard to isolate, they may be subject to genetic injuries of the aging process. Not all different organs have these present. So for example, it doesn't appear that the pancreas has stem cells that are capable of making the insulin-producing cells. And they may senesce, they may age and just disappear. They have been used as replacement therapy for more than just blood cell development. There is a whole area that's being developed of possibly contributing them, using them to try to overcome the defects associated with a ischemic injury to the heart. This is a controversial area, but one that's growing, particularly in Europe. And then I just wanted to point out one other area. Am I running too short of time here, David? I should stop. I will stop. 2 minutes. Okay, let me talk about cloning, just so you know what it's about. Okay. So this is, I think, important because it's a very distinct type of embryonic stem cell. It's important to us as we think about how to use these in medicine because it is a way to create cell types that are representative of disease model, of diseases. So unlike the products from a fertility clinic where the people going in and donating are normal, here we're talking about being able to create embryonic stem cell lines that are from people with illness. So, and I'll mention the advantage of that. The way this works is basically you take an unfertilized egg, you remove the nucleus, and this is a process the way it looks in the laboratory. So this cell no longer has its brains, it just has this sack of protein contents that are very powerful because you can now take a cell from an individual. Say this person has Lou Gehrig's disease, ALS. You could take the nucleus of one of their cells, theoretically a skin cell, transfer it into the egg, and the egg then has the ability to actually reprogram this nucleus. So this nucleus forgets that it's actually a skin cell and starts to perform as if it's an embryo and can actually create a structure that is this blastocyst. Now, this is then capable of possibly producing ES cell lines, embryonic stem cell lines, and these, because they have all of the different genetic components that possibly led to this person's disease, now allow us to generate models of those diseases. So, for example, if this is a neurologic disease like ALS or Lou Gehrig's disease, you could then theoretically create nerve cells in the laboratory that have all of the different complex genes that contribute to this disorder. That would allow you then to both look at why this disorder develops and possibly now use it in drug screening to identify new strategies to possibly prevent or treat this disorder. It also allows you to not just create models of disease, but as you might imagine, this now has the immunologic content that is identical to the original donor, and so you could possibly make custom cell lines. Anyway, all of this is a lead into just the idea that stem cells have many different uses. You'll hear more about their potential use as cell therapies, not just as individual units, but also in complex three-dimensional structures that the bioengineers in the audience will be telling you more about. So, thank you.
Let's take the next step, and that is how are we going to use the cells and other types of cells of similar nature that David talked about and begin to construct a tissue. And the next step to doing this is to think about how to place them on some kind of substrate or scaffold or material that allows the cells to do their job. And if you think about the cells in your own body, with some exceptions, particularly your blood cells, almost every cell in your body needs sort of a substrate or a scaffold upon which it has to sit and do its job if it's going to function. And so part of the tissue engineering paradigm is to make sure you provide a suitable environment for a cell so it will create the tissue type that you want under the settings you create for it. So in thinking about what I wanted to tell you today, I said, well, let's pick one of the biomaterials that our group studies extensively, and that is the material called silk, which many of you are, I presume, familiar with from the textile industry. We work on a number of different biomaterials, but I thought this one told the best story, at least for this particular audience. So I take you back to 200 BC to tell my story. And the story starts, for those of you aware of this history, really the trade route that developed way back in history between East and West. And this is really the origins of many aspects of interchange. So we could call it the information highway, if you will, before there was modern day information highway. In fact, the process of growing silk, which is called sericulture, which is really domesticated silkworm culturing to make this interesting fiber, protein fiber that we're going to talk about, goes back at least 5,000 years. So it's extremely old, domesticated process that was developed. And it was such a high-valued item that it really did form one of the backbones of many reasons for this trade route that developed between East and West many years ago. There were many other considerations, obviously the political, religious, and economic inroads that led to exchanges of religion and so on. But in reading about this historically, which I honestly hadn't done until recently to get together this slide for the talk, you see quotes in the historical literature that talk about cultures craving silk. And so it's hard for us to imagine today, except for the students in my own lab because they study it. But literally, the cultures would crave silk to the extent that it was more valuable in some cases than gold. That's how valuable this commodity was, for many reasons. It was a unique material at the time, only present in the East. The West had never seen it before. It felt nice. It formed these beautiful textiles that only the royal families could use and so on. So there were many reasons, supply and demand, I suppose, at the time, drove this kind of culture. In reading about craving of silk, let's bring us to the present day, which is craving of caffeine in this society. And I think we can all relate to how they must have felt back then, but I can certainly relate to that. Kelly, that one's for you. So anyway, let's move from that historical perspective to today. And today, David spent a lot of time in the first talk talking about the bottom half of this slide, which are all the nice cells that populate all the tissues and organs in your body. What I want to make sure is clear to you in my talk is that these cells don't do much in and of themselves unless they're communicating with the outside world. And in my book, that means all the materials that surround all of your cells that form what we call in science the extracellular matrix. Just think of it as a scaffold or a rector set or whatever that surrounds all of the cells that make up the tissues and organs in your body. And these materials must do everything for those cells. They must take whatever genetic program the cells activate in response to the environment and in turn reciprocate that information. So, for example, if you get sick and have a wound or a cut, the cells have to respond and create new tissue around themselves so you heal properly. If you are exposed to different mechanical forces, the cells feel that, respond, change the nature of the material around themselves, and allow you to function in the same way. So there are many examples of how cells, through their membrane, their interface, interact with those surrounding materials and allow all the normal functions that your tissues and organs need to accommodate for you to live and breathe and function. So that takes us to designer biomaterials, which is what I wanted to briefly tell you about. And that is, what do we need to do then with the materials we create in the laboratory to make sure we're able to do the best job possible to direct the stem cells that David talked about towards a specific outcome. And you'll hear from Gordana later about growing heart tissue, so there are different material design considerations for that versus growing bone or cartilage and so on. So there are lots of things we could say here. Just briefly, you clearly want materials that the body won't reject outright, so they have to be reasonably biocompatible. You want materials that are what I call versatile, so you can make lots of different versions of the materials by taking the particular polymer or other material that you're interested in. You want to control how quickly those materials might disappear once you place them in the laboratory or in your own body. In some cases, you want them to stick around a long time. In other cases, you want to get them out of the way quickly and let the body take over. Again, we want materials that are very accommodating in terms of being able to be decorated, hang things on them so they can direct the cells even further. And clearly, and many of the reasons why we're talking about the Silk Road today is because of the mechanical properties. In many cases, and for that matter, most cases, almost every tissue in your body has to accommodate a range of mechanical forces. And therefore, if we're going to create a designer material, we need to try and mimic or at least approximate the kinds of mechanics that your body normally sees and feels if the cells that we're growing on these materials are going to behave in the way we would like them to behave. So that takes us now to the silk sort of issue and why we have studied silk so extensively in our research group. And briefly for silk, for those of you not familiar with it, this is the age-old process of sericulture from the Silk Road we talked about. And here's the worm. It begins to decide to turn into a moth and begins to spin around itself a cocoon. And this is basically pure protein, our biomaterial in this case, that it creates an encasement around itself. Here's a lot of them. So it can survive the transition from a worm to a moth state. In the process of sericulture, the domesticated process to produce silk, this process is simply interrupted. The worm is not allowed to emerge as a moth, obviously. The cocoons are essentially boiled, the strands of pure protein removed, and spun and used in the textile industry. So that's the origins. There is a whole new world of silk out there besides just the domesticated silkworm, however, and that comes from spiders. And so we and others have extensively studied the origins of these beautiful engineered structures you would know as orb webs that you probably normally get out of the way because you probably don't like them. But the spider is really a remarkable bioengineer in and of itself because for very, very small amounts of material, it creates a very, very large net, which is optimized in terms of mechanical properties and function to catch flying insects of just the right size, so they get entrapped and then can be consumed for sustenance, but not to catch anything too big like a bird, let it go through and not catch it, and not have it serve as a trampoline and have an insect bounce right back off and lose that captured prey. So we and others study extensively some of these silks as well. The unusual feature of a spider is it actually spins many, many different kinds of silks, whereas the silkworm spins only one kind of silk. And each of these silks has a different function. Some are for the structural members of the web. Some are sticky to hold on to surfaces. Some are also to make cocoons and so on. So we can learn a lot from nature. We can borrow designs, protein designs, and use these to generate new kinds of designer biomaterials. In the lab, we spent a lot of time figuring out how silkworms and spiders carry out this process. And again, for today, I'll just allude to the fact that whether we're talking about a silkworm that spins actually from modified salivary glands in its mouth, out of its mouth to make the cocoons, or the spider, as we just talked about, that has these beautiful spinnerets in its abdomen to make the orb web, all of this happens in glands within these animals. All of it happens in a pure water system, and the proteins that we're talking about are very, very they do not like water very much. And yet these animals have solved the process of processing into very, very nice, mechanically robust fibers over eons of evolutionary control of protein chemistry design, which, again, we would be happy to talk about after or later tonight, if you would like. Ultimately, then, we are stuck, if we stick with the Silk Road, with a single silk, the silkworm silk. And so our goal has been to morph this material into a whole new range of useful designer materials that we can use for the tissue engineering paradigm. And so we can take, for example, cocoons. We can extract out the sticky parts that hold the cocoon together and just get pure fiber. And we can go through a laborious process to take this fiber and bring it back into pure water solutions.
Good evening. I'm really delighted to be here again in the room that has definitely the best view in Boston. And you are all very kind looking at us instead looking back. So when David started to organize this event, he came over and he said, okay, you'll talk about heart tissue engineering. I said, all right. And then he gave me a title that was really too radical, even for him. It had something about Valentine's Day, and I'll come back to this on my last slide. So this was rejected. So we sort of settled that semi-scientific, semi-popular title that's about the growth of beating heart tissue in lab, and the eternal question, can this tissue mend a broken heart? I mean, literally broken heart. Broken heart. So I'll start, and I forgot to say, another condition was we cannot see each other's talks. We always compare our talks, but he said, no, this time we're done. So it's a coincidence that actually we started about the same time. I mean, I beat him by 100 years, if I remember correctly. So I would like to walk, given that we have even kids of our colleagues here who don't know too much about cardiology. And we have a colleague who is distinguished cardiologist, and so like out of respect for the audience, let's just walk through a couple events, milestones in cardiology to put things into the context. The first message is everything started very long ago. And you can find in literature, if you look for it, evidence that there was a cardiac surgeon in China who used anesthetic 300 years before AD to do some interventions on the hearts of people, which sounds like really remarkable. And then there is a very long break. Not much happened for about 2,000 years, and then things started to evolve. And there are very many events that you may know, like I'll just say that blood circulation was described about 400 years ago. EKG was discovered just about 100 years ago. Transplantations started actually in 60s, and there were several attempts to repair terminally diseased hearts by transplanting either chimp heart into the human, and then the revolutionary surgery in 67, Christian Bernard, who did the first human-to-human surgery. And then some other important developments, like the use of the artificial heart, the very famous Jarvik heart, that was capable of supporting people's heart function while they were waiting for transplant. And something very important in 1980s, which is actually the cyclosporine. That's the first drug that really efficiently prevents rejection. So today, we have a situation that there are very many people that have other person's heart. So what's wrong about this? I mean, what's wrong about continuing with the existing therapies? So here is a little cartoon that tells you what we have available, and I'll just say briefly what we use and when. Obviously, there are drugs, the first choice whenever possible. There are artificial devices that never work well on a long term, because we just cannot make heart out of plastic material or metal. It doesn't work. Body rejects it. There are transplants, and they are very seriously limited by the lack of donor organs, obviously. And unfortunately, most people die on waiting lists rather than from the surgery. Surgery is very successful these days, but there is not enough organs. And so we come into two therapies that are actually experimental treatment modalities that are currently under research. One is, you probably all heard about ideas to inject various types of cells into the heart or into the circulation to come to the heart. These cells range from just normal muscle cells, say taken from a leg of the patient, to adult stem cells that David Scudden described that can be obtained either from fat tissue or bone marrow, to embryonic stem cells. And there were quite a few reports, actually, that this helps to some extent for the reason that we don't understand very well. We don't know, is it like better revascularization? Is it some kind of rejuvenation of the heart? What it is, we don't know, but there is some hope that this may help. There is also a major problem about cells not staying put where you inject them. So the cells stay there for a little while and then they go. Alternatively, we are talking about something that's shown here in a very extreme variation, which is let's take out the piece of tissue in the heart which is defective and replace it with a piece of healthy tissue. And this healthy tissue is actually the substitute I'll try to describe to you today, something that we would ideally grow in culture in the lab, for the reason that this is then an untapped source of tissue. You can grow as much tissue as you like in the laboratory. So the shortage would be really addressed. So essentially, the drivers of cardiac tissue engineering, the reason why many groups are very actively working in this area are many, and I try to summarize 4 of them that I think are the most important ones. One is heart tissue does not regenerate. The ability is so minimal that we cannot rely on it. So if there is an infarction or any disease, the heart tissue pretty much stays at the same level. It doesn't repair, self-repair. heart failure and heart disease in general kills more people today in this country and the Western world than all cancer combined. So this is like a big problem. Scarcity of donor tissue that I mentioned is very serious. In the year 2003, it was 3 to 1. So only 1/3 of people that really needed heart transplant could have one. And finally, engineered tissues, as I'll show you a little, can serve as models for biological research. And you see articles like this one that I quoted here, Goodbye Flat Biology, written by biologists. I mean, this particular article was in a Nature magazine, written by cancer biologists. So they are advocating for the use of more realistic models for fundamental biological research. Now, the idea of tissue engineering, this is a very old slide that's extensively used, and I will just use it to show you what is the key idea. We use cells, and cells are the actual tissue engineer. So everything done in terms of tissue engineering is really done by the cell, the true architect of the tissue. And then we have two components that we use to help the cell to do the job the cell is supposed to do, which is to rebuild the tissue. One is a biomaterial scaffold. So David Scudden described some of the cells. David Kaplan described biomaterial scaffolds. So we place the cells onto the scaffold, and then we culture them for a period of time. This is the part that I'll describe to you. And if everything is done right, then we obtain something that resembles normal tissue, and then it can be implanted to replace diseased or missing tissue. So this is a concept. So the concept is pretty simple. You take the cells, and then you make the cells. You somehow trick them to make a tissue, then you use this tissue for clinical. So how does it happen? This is a set of two slides, which are probably the most important in this story, and they pretty much echo the theme of our conference that we are holding these 3 days, which is we are trying to learn from nature, and by learning useful things from nature, apply this in laboratories to perform tissue engineering using cells. So what happens in nature? As you already learned a little, stem cells first grow, they multiply themselves, they grow in number, and they also decide which way to go. So this is the process called differentiation. So they become like a bone cell, skin cell, blood vessel cell, whatever. And then when the cells undergo this process of becoming specific, doing a specific job, then they assemble into tissue structures. In most cases containing more than one cell type, like some blood vessels, some nerves, some specific cells. So during both steps, there are several things that are important. First of all, cells need a template, a scaffold to attach to. They cannot function in vacuum or in suspension. They need physiologic conditions, for example, body temperature, body pH, and other conditions. They need a number of regulatory molecules. You all heard about growth factors and similar molecules that really determine what will happen here. And they also depend a lot on physical signals, because our body is not static. There are electrical, mechanical, flow signals, and other signal cells are exposed to. So we would imagine that this all is important in this process. This is very complicated. So it's complicated for many reasons. First of all, there are multiple cues, multiple signals. It's not one factor that's dominant. They act differently at different length scales. So something has a specific effect on a molecule, and then a different on a cell, and different on a whole organ. So it's complicated. Hardly anything is uniform in nature. So you have these gradients of properties. Timing is important. Some factors are important early, some factors late, and finally everything interacts with everything. So we try to lift some recipes from biology. We try to learn whatever we can learn about the conditions that are favorable for a specific processes. And what we do is actually we recapitulate these conditions in laboratory. So here is the architect of the tissue. There is a scaffold, which may be silk or another material. We place cells onto the scaffold, put them into an environment, which we call bioreactor, which is essentially like a substitute body, something that does the function of the body. And then in the case that I'll be discussing today, we obtain a little patch of tissue that will eventually is envisioned to patch over like a Band-Aid over an infarcted area on the heart, or be used in laboratory for two things. One is to go back onto the previous slide and try to learn a little bit more about how things really happen in biology, like what are the sequences of events, and also to do clinical studies, such as, for example, drug screening. So this is what we do in tissue engineering, and I'll really talk only about heart. Heart is a true marvel of nature and engineering. I mean, it's an amazing organ. It has about 3 billion cells, adult heart. It beats about 3 billion times in our lifetime. This is a lot. Every day, heart pumps about 7,000 liters of blood. If you think in gallons, I mean, this is about 2,000 gallons. It's a lot. And through 100,000 miles of blood vessels. So these cells are very hard workers. It's truly remarkable. And if you look in little more detail, what makes heart special and what makes our task to engineer anything heart-like so difficult, there are essentially three things. I would just point at three things. This is the actual photograph, like a high magnification photograph of the human heart tissue. So this here is a capillary, and it's about 10 micrometers. So it's 1,000th part of the millimeter. So it's a very small. And this is the heart cell. And you can appreciate that the heart cells are very densely packed. And essentially, each of these heart muscle cells is flanked between two capillaries. Nature doesn't do anything without the reason. And the reason is that these cells that are working very hard, they need a lot of oxygen. So if you look into the vasculature of the heart, you see it's very, very dense. So the first lesson to take from biology is we need to provide oxygen to these cells. And we cannot even think about fooling the cells that they can live without less oxygen. They just die if you don't give them enough oxygen. So this is the first big task. The second important task is if you look into how these cells work, very simply stated, there are electrical signals that come from so-called pacer cells, cells that are actually generating the heartbeat. They spread over the heart cells, and then they beat in sync. So the heart responds to electrical signaling by beating, by twitching. And this is what pumps the blood. So this feature, which is called coupling between electrical excitation and contraction, is something that we need to resemble somehow in culture if we really want to keep these cells happy and healthy. And I will just show you two things that we do in laboratory that result in heart-like tissue that is beating and has some of the structure of the heart tissue. So essentially all our science is summarized in this and the next slide. So to provide enough oxygen to the tissue, so what we do is we take a biomaterial scaffold that's very porous. It's one of these scaffolds that David showed, similar to that. It's mostly void volume like a foam, and then we poke channels through this scaffold. So you obtain a scaffold, which is shown here on the picture, which is pretty much like mimics to some extent vascularized structure. So this will be some kind of artificial capillaries. We seed the cells into this sponge-like structure, and then we flow culture medium, which contains sugars and proteins and everything else that the cells need, through these channels, so that there are very short distances to the cells for oxygen supply. And to even further assist the oxygen supply, we add oxygen carriers, perfluorocarbons, if you ever heard of them, which to some extent mimic the role of hemoglobin in our blood. So they carry a lot of oxygen. So essentially with this kind of cultivation, and you see just one channel here and everything is filled with the cells, we are able to support cultivation of physiologic cell density, physiologically high cell density. The other thing that we do, which is shown here in a pretty artistic way is actually that these tissue constructs, these polymers where we put the cells on, we stimulate during cultivation, during the culture, using regular cardiac pacemaker stimulator that's used to actually stimulate the human heart. And the idea is actually, so this is like a picture of a large construct that shrinks as it beats, and then the idea is actually to have a patch that's functionally contractile and to lay it over the heart. So this piece of tissue actually has, if you look into what we engineer in the lab compared to native, you see resemblance of the structure here and here. And essentially, what you see here blue are the nuclei of the cells so that you can be oriented. And the green stuff is actually communication protein. So this is the protein, if present, that shows us that the cells are connected to each other and they can transmit electrical signals. And you see that this resembles the lab-made structure, the native structure, relatively closely. And importantly, this is after only 1 week of culture. So this is very fast, very short culture. When you look on the other magnification, the other part of the cell, so this was communication, the contractile apparatus. Again, this is engineered and this is native. And even if you don't know how to read this kind of a slide, you can see that the structures that you see in the engineered tissues that are called striations are pretty similar to the structures that you see in native tissue. Relatively recently, we also started to look into implantation of these constructs, and the only thing that we have done so far was to look if we can get vascularization in vivo, which means functional blood vessels. And what you see on this little image, blue are the cells again, green is the marker, but actually these cells here in the center are actually blood cells that show you that something is flowing through these channels. And essentially the idea now, all this that I showed you was done with animal cells that are our experimental model. We are now trying to move towards the use of some kind of human cells, again with the idea to somehow repair this damaged heart muscle and to develop an alternative to this therapy, which is essentially injection of the cells, which is not completely controlled. And let me just explain what we are doing now. So we are making the baby steps towards using of adult human stem cells, both from bone marrow and fat, and embryonic human stem cells, and trying to coach these cells towards going into first becoming cardiac cells. So this is the first and very difficult step, and then fusing into the tissue like we have achieved with the animal cells. And we really don't know which of these will work best. Our strategy is to use these stem cells, to use advanced tissue engineering systems, essentially to apply all and any methods that we know that work with animal cells to human cells and see if it will work, to go towards obtaining vascularized heart muscle, because this is the only one that will work, and then to show using rigorous animal models, which is a patch over the heart, that they work in vivo. So we would like to obtain this consisting of human cells. And this is my last slide, and coming back to the title of my talk, our only product recently was actually this web page at MIT. So we got a Valentine's Day web page, which was very cute with these letters MIT. So this is like 3 mended hearts. And this was actually related to an article that we published about engineers aiming to mend broken hearts. And this is another artistic vision from a paper where actually someone made like a scaffold. So there is a scaffold around the heart, and then there is someone putting heart. So essentially, to summarize in a more serious way, we feel that there is a huge hope and huge potential, but in all practical terms, we are really at the end of the beginning. So we showed some results, but there is a very long way to go. I'm not sure if it's as long as Silk Road, but actually there is a long way to go until any of this becomes clinical reality.
when I was preparing this talk a couple of days ago, and I asked my kid, my son, I said, well, what should I talk about? And he gave me two advice. He said, one is make sure it's funny. Two, make sure the slides are colorful. Slides are colorful, but I don't think it's going to be funny. I'll let you handle the jokes. And my two kids are right there, so it makes that's my biggest challenge tonight, to make sure they are satisfied with the presentation. And you have heard about the cell source, which is very important for tissue engineering. You did hear about the biomaterials because these cells don't live in vacuum. They attach to these surfaces. And you did hear about the signals and different elements that are important when they interact with each other. How do you build bioreactors and go to a three-dimensional structure? So my job here is to talk a little bit more about, drill down a little bit more into the manufacturing. How do we make, what are the different options, and give you a sense of different organs that have been built in the laboratory. Some are very early stage, some are a little bit further advanced, and give you a sense how engineering type tools and technologies can impact building or bioengineering of tissues. The reason for this is not clear to all of you. You're all experts in tissue engineering by now. Basically, there's a huge shortage of organs, and many of our organs fail, sometimes early in life, sometimes late in life, but we cannot rely on that fact that they're going to be working forever. And when they fail, we need alternatives other than transplantation because there are not enough organs to transplant for these problems. And that's really the major motivation for us to work in tissue engineering. Here is how engineers work, and it's like construction engineering. We build tissues, and this slide actually has several important messages. One is that we may have to build these things one at a time. You have to realize that your heart did not form overnight. It took a whole development process. It took many years to form that heart. It started from an egg and a sperm and divided into little cells and formed an embryo and then went into developmental stages. So it's brick by brick. On the other hand, when your organ fails, tissue engineers like to build it overnight, create an organ. So there's an element, how do we build it? How do we bring the different elements, cells and materials together to create a functional organ is very important. And also the vasculature is very important because when you build these organs, you need to be able to bring in blood and supply nutrients and remove the waste product from this. So from a construction worker perspective, it's a very challenging goal. And one of the things is organs, although they are big when you look at them from outside, at the micro scale, they are very small. And controlling the making of microstructures is very important in our ability to build organs. And here you see length scale going from nanometer to about a meter. This is 1 millimeter. Your hair will be about here, 100 micron. This is 1 micrometer. And people actually have built micro devices, and we have put viral particles on a nanotube built by human beings. We can build guitars that are about a few micron. If you have very tiny hands, you can play that guitar. And it's more challenging than tissue engineering, but it's doable. And here is a mechanical gear, actually, that is the size of it is about a micron. And very impressive. You can build mirrors. We can move these things, actuate these things, and we can also integrate them into a circuit, so create complex structures. So we can build small things, and then we can make them complex. And why is this so important for biology? Well, many of the length scales that we deal in biology really overlap with the length scales that are important in micromachine systems. Here is a viral particle, is right here. This is a protein. Here you see the nucleus of a cell, which is about a micron. Here are cells, mammalian cells, individual cells. These are about 10 micron here. And then here you see a liver lobule, a sinusoid, which is about 0.5 millimeter to 1 millimeter in size. And so it goes from simple things to an organized structure with blood vessels, in a sense similar to integrated circuits with all the different parts and elements. And the guy who made a major impact in this field, not in tissue engineering, he probably didn't think about tissue engineering when he was giving his famous presentation on There is Plenty of Room at the Bottom in 1959, Richard Feynman. And but he thinks that he was thinking at the time has not having impact in fields in biology and medicine. And his notion there was can we build smaller and smaller things? So the ability to control things at that length scale is, of course, important for biological systems as well. How do we do that? We start with something that's called a wafer. These could be 12-inch wafers, and it's a silicon-based material, and we can create the structures on this wafer, and you do it in a clean room. The reason why it's yellow, some of the chemicals we use are very sensitive to light. These are standard clean rooms that are used to make integrated circuits. This specific one is at Massachusetts General Hospital. As far as I know, the only clean room in the country that is in a hospital, and where we can make these integrated circuits. And you have to wear these funny suits and to be able to work in this environment, because even a dust particle will ruin your manufacturing. And the way it works is you take that silicon wafer, put a photoresist on it, you shine light through a mask, and you only open certain areas of that mask, areas that are exposed to light and chemically removed, so you can create these structures. You can etch it, and you can etch it in different ways. So the message I'm trying to give you is that the integrated circuit technologies, microsystem electronic technologies that we are identically using those techniques in tissue engineering, as you will see in the next several slides. The ability to control at micron scale structure is clearly important, as you have heard from the prior presentations, that cells live at this length scale. The ability to control the chemistry on this surface is also very important. Now let's look at one example where we use this sort of approaches to build engineered tissues. This is an electron micrograph of a liver. This is not engineered, this is real liver. The length scale, this is a liver cell called hepatocyte. Those are the functioning cells, about 10, 15 micron this way. And there are these other sinusoidal or mesenchymal cells. The interaction between these two cells is extremely important. And here are the blood cells, here, red blood cells. It's a very porous structure, and it interacts with its environment in a very specific way. And the way these cells interact with the other cells, mesenchymal cells and hepatocytes, is extremely important. Using these microelectronics techniques, we can put zip codes, so to speak, on these surfaces, where we can actually put hepatocytes or any cell type at any location on the surface, create these sinusoid-like structures you have seen in the prior slide, which is the real liver. And these are individual hepatocytes that are lined up, and you can create this complex structure pattern cellular systems as a beginning of building organs in the laboratory. These cells like that will not function. Hepatocytes alone like this won't function. They are alive, but they are not going to function. They don't have the right cues and signals from their environment to function. They're just sitting there alive. At some point, they'll probably get bored and die. And so you need to create the right environment. One of the right environment here was the other cells that surround them. Before, we thought these mesenchymal cells were so only functional from a mechanical perspective, but now we realize that these two cells talk to each other, and there are very complex signals that go from this cell to that cell and causes them to differentiate. So creation of more complex tissues, such as this one, which is a co-culture of hepatocytes, which are in red, stained with a vital dye, so and then you see these structures around them green. These are actually mesenchymal cells that are lined up, almost similar geometry to the liver sinusoid, where each of these mesenchymal cells are interacting with the neighboring hepatocytes. When you do that, these hepatocytes immediately become functional. They give the term, fancy term for that, differentiate, and they gain their phenotype, normal phenotype, and these cells now have been shown to function at levels similar to normal physiological levels for months. So it's a way we as tissue engineers create structured organs using microsystems engineering that are functioning. Now, once you have this, what do you do with it? One of the things we would like to do is take it to the next level, build actual organs that we can use for clinical purposes. In the case of liver, there are two major fundamental problems. One is acute liver failure, which happens because you ate, and it's primarily children suffer from that, either from a Tylenol overdose or mushroom or some toxic overdose. The liver will fail. This liver will have full capacity to regenerate. If the underlying disease is treated, that person will survive. And it will be like they had cold, they're treated, cured, they can go home. Right now, the only modality for these patients is organ transplantation, which will be not economically very expensive, $200,000. But the worst part is that these kids need to be immunosuppressed for the rest of their life, and a major compromise in quality of life and safety. And so the ability to have, in this case, the tissue engineering is, you're looking at it as a temporary support of a failing liver until the underlying disease is. So the issue here is an extracorporeal device. It's temporary. You need it probably for several weeks. And the way this could be built, you take the blood, you separate plasma from blood cells, put the plasma through a liver bioreactor that has these micropattern cells that are fully functional cells at the bottom. You need about 10% of your liver. That will be about 10 square meter surface area. So these are going from micro scale to macro scale. Very challenging engineering issues. 10 meters square is a big surface area, so you need to make it in multiple layers, laminated, and you need to make it in a micro scale where the total volume of this extracorporeal device is not so big so that you will actually kill the patient. It needs to be less than certain amount, several liters for this device to work. In this case, so these are actually tested in animal studies and moving forward, but the concept here is acute liver failure, temporary device. You get there, no time to build it. This is not going to be organogenesis. You need it right there and then and hook it to the patient. To take it to the next level, you need to build a vasculature. In the case of chronic liver failure or other cases such as diabetes and pancreas, where you need an implantable device, not a temporary device, then you need a vasculature. And here you see the vasculature. I believe this is from an islet, not a liver, but it's very similar to liver vasculature. Extremely vascular organ. And you need to be able to build this vasculature using tissue engineering tools. One way to do that, again, using these microelectronics type tools. This is Jay Vacanti's slide from Mass General. The idea here really to build a microvasculature where the main branch comes here, it divides into multiple layers and then it creates these arterioles and then collected and the vein comes out of here.